So although this is not the most basic of molecular biotechnologies, an important technology is in fact something called gene cloning. And so this is just an overview. I'll show additional ones that gives you a sense of what gene cloning is all about. So what is gene cloning? It's taking DNA or genes or pieces of nucleic acids that are associated with one organism and making them a part of a different organism. In fact, this little bit of blue that probably is not visible from way out there is a piece of, in this case, DNA that came from that organism. So how do we gene clone? What we do is we first take a cell, or in fact a virus, and we have to isolate the genetic material. If it's DNA, we're fine. If it's RNA, it has to be converted into DNA. We clean this stuff up, and then we cut the DNA. The way we cut the DNA can vary, and we'll go into one means by which we cut the DNA. But then that DNA is recombined. It's actually inserted, is the word people use, into what is known as a cloning vector. And that cloning vector, typically it's something called a plasmid. In fact, this is a plasmid. Little piece of circular, in this case, DNA that's found as an extra chromosomal piece of hereditary material, in this case in a bacterium. We put the piece of DNA that we want to clone into a cloning vector. We have to make sure that it's attached to that cloning vector. It's ligated to it. And at this point, we have recombinant DNA. We don't have a recombinant organism yet because that DNA is sitting in a test tube. So the next step is to have various means by which we take this DNA and we shove it into a recipient cell. Transformation is the uptake of naked DNA, DNA that's not associated with any proteins or anything. And transduction is the uptake of DNA that's associated with viruses. In fact, in gene therapy, we often use viruses as a means of getting new genes into cells, such as the cells found in a grown human being. But anyway, we have to have some means by which we get the DNA inside of the cell. Once we get the DNA inside the cell, we have what is known as a transformant. And then we can grow up the cells that contain this new DNA and thereby amplify that DNA. Before PCR, this was how DNA was amplified in small bits. And also, once we have the DNA cloned, we can manipulate that clone molecularly, modifying it so it does novel things. But these are clones. When we clone DNA, when we genetically modify organisms, it means that we've taken DNA from some other organism, and in some capacity, we've inserted it into the genome of a recipient organism. Often that insertion occurs extrachromosomally, but not always. There are certainly many technologies that allow you to insert the genes directly into the chromosomes of a recipient organism.